All right, good morning, everybody. Happy Friday. Woohoo.
Morning.
It seems like it's taken a long time to get here. All right, so today we will start Mendelian genetics. That's what's on the list for today. Let's get the chat up. And first up, before we start talking about this stuff, does anybody have any questions about what we covered on Monday and Wednesday? Let's give you opportunity to go through anything that didn't quite make sense or needs clarifying. You can have a little bit of a puppy shot in the meantime. Fortunately, the other one's off at the vet's today because she's having an operation. So no cute and fluffy little one, unfortunately. Okay, so what we're going to start today is a pretty decent run at transmission genetics, right? So let's just check the schedule, see all the stuff that we have in there. So this is going to be the kind of like the classic start, I guess, to transmission genetics, talking about Mendel and Mendelian genetics. And really that's useful as a framework and to kind of learn the ropes. Then we're going to be looking at some probability and statistics, right? So a lot of transmission genetics is math, right? Logic and math, basically. You know, what do I expect to find? And essentially, does what I find match what I expect, right? So there's a bunch of math and statistics in that. And that's a really useful way of kind of thinking about what's going on as well. And then we got some cell division, which is pretty important to understand the kind of the underlying basis of some of the stuff that we're looking at in terms of transmission genetics. And then we have a whole section, pretty much 2/3, half of the next section, which is going to be on non-Mendelian inheritance. Right, so the real purpose of what we're going to be doing up until exam 1 is really getting the basics in, right? So that when we get to non-Mendelian inheritance, which is the really interesting stuff, then we're able to kind of really go to town on that, you know, properly understand what's going on. Oh, I just noticed that should be a 3. All right, so oh, hang on, questions. Question about printing off the homework. Not that I know of, other than simply taking screenshots. I don't really have a way of downloading that as a text, unfortunately. Sorry about that. Yeah, so just take screenshots really, so you can use those later on. Bianca, I don't think there's any reading associated with the probability and statistics because that's a kind of a module that Dr. Page, who taught this course before, developed. And really the kind of the reason why he did that was because, you know, math is important in genetics. You just can't get away from it, unfortunately. And it's a kind of known area of weakness of students is dealing with math. So he kind of just developed that chunk and is like, here you go. here's all the math that you need to know for what we're doing, or math and statistics. Obviously, there's a lot more to it than that, and that's why I think some of you are actually doing it right now, but there's a course that he now teaches called Biostatistics in something and something, can't remember the rest of it. And you'll get a very thorough covering of all of the math and statistics needed for biology, which is a worthwhile course. Not personally my cup of tea, but that's because I'm crap at math. But it's very useful because I use the stuff that he teaches, you know, on a pretty regular basis. It's very important stuff to know. Oh yeah, yeah. That's a course that I'd probably fail, or at least, you know, be at the certainly the lower reaches of the class. Yes, statistics isn't my strong suit, to be honest. All right, so let's get cracking. So before I get into, you know, Mendel and stuff, I'm not super interested in kind of history of science, really, because science is generally a very forward-looking process, right? It's really about what we don't know and figuring out what that is. But some of it's really useful to kind of get in terms of context, but we'll get to that in a bit. What we're going to do first is something really dull and tedious. And we're going to go through some definitions. Yay, definitions. I guess, I don't know whether it's better to do definitions on a Monday or Friday morning, but either way. These are important words, right? So these are part of the language that we'll be using, right? And if you can't speak the language, you can't understand what we're talking about, right? And that applies doubly so for exams, because I'm going to be using very specific language and mean very specific things, right, in the exam, and you need to understand what that is, right? Otherwise, you'll be lost. But I don't think I've told you this little anecdote yet, but when I was living in Medellin, Colombia, way back ago, when I was but a young man, I didn't know how to speak Spanish. Well, my uncle taught me Spanish before going. He said, well, just add an O and an A to the end of every word, English word, and there you go, speaking Spanish, which turned out not to be very useful. So what I did was I was working in a lab in a research institute. I had like a little book, and as I learned new words, which at the beginning was, you know, constantly, I would just write them down, and every evening I'd go back over them. And so over time, you know, my vocabulary kind of expanded. I learned the basics of Spanish, and I could actually end up speaking pretty reasonable Spanish. Not fluent, unfortunately. Wasn't there for long enough, but pretty good. And so I'd really suggest something similar for you all as well, right? To write down in your notes particular definitions that you're not familiar with. And those need to be part of your revision when you go back over your notes. Do I know what that means? Yes or no? No? Learn it and just keep doing that. And over time, you'll find that all of this vocabulary is just second nature, right? Anyway, so there's a very particular way we talk about stuff, and there's a very particular kind of logic to how these words work. Right? The basics are the gene, the allele, and the locus. Right? So locus is really where a gene is at, right? It's physical location on a chromosome or in your genome. Gene is obviously the, I mean, this hereditary determinant of a trait. That's very true, and particularly applies in transmission genetics. It's also the kind of unit of information, right? The bit of DNA which encodes something which does something, right? Essentially is the determinant of a trait. Now, alleles, this is something that people get tripped up on a little bit. An allele is a different version of that same gene. So I have, well, I actually don't know, I might have 2 different alleles for eye color. Most likely, well, I have many genes for eye color. Shit, that's a really crappy example. Yeah, let's say asthma, right? So I have, or used to have asthma. So for any particular gene that controls that, I most likely had a particular allele of that gene, which resulted in me having asthma. Now, some of you that, or most of you that don't have asthma will have the same gene, right? We all have the same genes, all of us. I think it'd be very, very weird if we didn't. Oh, I'll get to that in a second, Jake. But we all have different versions of that gene. Now, they didn't necessarily, hang on, Rebecca, I'll get to you in a second, Jake. Rebecca, no, locus is where a gene is located. So where a gene is expressed, that's a different question, because then you're asking about, you could be asking about where in the genome it's expressed, which isn't really how we would think about it. But if someone asked me, where is a gene expressed, I'd be thinking about location, like what cells is it expressed in, what tissues, what context, things like that. So locus is just its physical location. It's like a, like a mailbox with a house number on it, essentially. Right, that's it, just there. Let me just finish everybody on alleles, and then I'll get back to the questions. So an allele is simply a difference in the sequence of a gene. Now that difference may have a consequence. It may result in that gene not functioning, or that protein not working, or all kinds of different variations on that thing. Or it could have absolutely no effect whatsoever. If it is a difference, it is an allele, right? So that's something that's really worth kind of banging home. Questions from the chat. Yeah, that's an interesting one, Jake, about growing out of asthma. So part of that might have been, I think it was actually, so it was childhood asthma until I went to university. And I'm just trying to think how to put it. Basically, I think it was about being exposed to, what the hell are you doing? Go away. Being exposed to a broader array of immune challenges, essentially, right? So asthma is essentially an immune overreaction is probably the best way of putting it. And there's other versions of that too. And one reason could be that your immune system is just kind of like bored and twiddling its thumbs and wants something to do. And so if you give it something to do, and actually I think taking up mountain biking and having mud thrown into my mouth on a weekly basis was part of that, kind of diverts its attention away from asthma. That's kind of not so much desensitized, but more it was the idle capacity of my immune system was taken up by something else, I think is probably the best way of putting it. And also being exposed to lots of other people, different environments, things like that. In terms of vision, Berenice, so vision is a complex trait. There's an awful lot of genes which control the development of the eye, different, like even eye color, for example, is controlled by multiple genes. But certainly the collection of alleles that you have for the genes that control vision is what determine what vision you have. That's probably the kind of the most comprehensive answer I could give. We don't really actually know all of the different genes that are involved in determining your vision, to be honest. It's an extraordinarily complex process. Just the development of the eye is a mind-blowing kind of deal. It really is. Going through puberty, actually, I had asthma until I was about 18. I still had it occasionally after that, cold-induced asthma, for example. But no, I think it was just the immunological challenge, essentially. right? Which I got from going to university and starting mountain biking. Okay, back to definitions. Ha! Oh, you've done a successful job of distracting me from dull definitions. Okay, so genotype and phenotype, also important. One thing to remember is that the genotype is the underlying basis of the phenotype. Right, so there are kind of two different ways you can look at this. The underlying basis of the phenotype. So the phenotype is what it is because of what collection of alleles you have, right? Which is your genotype. Also, you could think of it the other way, which is that the phenotype is the expression of the genotype. Right, so that's kind of looking at it from a different perspective. One thing to be very clear about, though, is that it works in this direction. Yeah. Yeah, kind of. Yeah, well, I'm not going to unwrap that analogy, Rebecca. So genotype equals phenotype, but phenotype, oops, if I can spell that right. Does not, that's kind of the best way I could do in typing it. Does not necessarily equal genotype. Right, so if you know the genotype, or if you know the underlying basis of a trait, and you know the genotype, you can say it will have this phenotype. If you have a phenotype, if it's really the dominant phenotype, you cannot tell what the genotype is, right? And that's because of the way in which dominance works. All right. Some more important words. Homozygous, two are the same. Heterozygous, each one different. All animals, not sure about, yeah, plants are kind of weird, but diploid, I think. Yeah, there's some weird stuff going on in Drosophila sometimes. But anyway, we're all diploid, and humans, at the very least, have 2 copies of each gene. Now, those 2 copies could be the same in terms of alleles. They could be different. Different, heterozygous, same, homozygous. Homozygous doesn't refer to what alleles you have, only that you have 2 of the same. Then we have, another thing is, sometimes we'll come across this, actually we've done already. Trait is the thing that you're looking at, e.g. hair color, oops. Disease, etc. Right. The phenotype is what version of that trait you have. So brown hair, cystic fibrosis, etc. Right. And the dominant phenotype is essentially the phenotype you have when you have at least one dominant allele. And what we mean by dominant allele, and we're not going to get too far into the weeds on this because there's a lot more to this than what we're going to cover. So dominant alleles are the ones that are expressed when you have at least one of them. I have to be careful with definitions, right? Because I can trip myself up. Typically, not always, typically the dominant allele equals the normal or functioning version. And that's often referred to as the wild type allele. So there's a few more kind of little wrinkles to those definitions. So when we talk about dominant allele, typically, not always, we're referring to the one that just is normal, right? So if the gene works, then that's the dominant allele. Now, there's lots of different shades of that. So this is really what we're doing is giving you the basics. There is a whole nother world of complexity, which, you know, hopefully you'll get to explore later on as part of another upper division course. And the recessive phenotype is obviously the one where you have two copies of the recessive allele. Where was I at? Do I have something that fell off of that? Sorry, got to change that. I'll upload a new version of this when I get off of here. What the hell? All right, these are all PowerPoints I inherited. So sometimes I come across things when I actually read them. I'm like, it's kind of weird. All right, now we've got some terminology that's specific to Mendelian genetics, right? When we talk about Mendelian genetics, we're talking about a very specific type of transmission genetics. It's a very formalized way of looking at inheritance. And it also inherently has a bunch of assumptions with it, which we'll get to in a little bit. So true breeding is a word that it's kind of old-fashioned in many ways. You know, you've probably, you know, some people like true breeding dogs. I don't particularly mind one way or another. All of ours are mutts. But true breeding basically means that all of the offspring from that dog, if it's mated with a sibling or another, you know, dog of its breed, will be the same. Now, if you do the same again, their offspring will be the same and ever and ever and ever more, right? And what it really means is there are no other alleles present in those individuals which will become expressed later, right, in other offspring, right? And so for that to be the case, those individuals have to be homozygous for whatever trait you're looking at. Now, obviously, they're not necessarily going to be homozygous for every trait, although there are, you know, you can buy mice, for example, for lab work, like CL57 black, for example. It is completely true breeding, right? It's very not. Oh, good question, Isaiah. Let me just finish this up. As far as I'm aware, you know, if you cross a CL57 black mouse to another one, it will only ever produce exactly the same. Oh, that's interesting, Rebecca, about King Charles Spaniels. So, yeah, a couple of things come up about true breeding dogs, which is interesting. One that King Charles Spaniels have problems because there were very few of them left after presumably bombing of England during the war. So that is what you'd call a population bottleneck, Rebecca. That's essentially where the original population kind of gets put through a funnel, and so what comes out is much more genetically homogeneous, right? And so unfortunately, if some allele that causes disease goes through that bottleneck, many more of the individuals out like at the bottom of that kind of hourglass that come out through that bottleneck will have that disease. That's also why, for example, kind of distinct population groups that tend not to outbreed in the US, for example, like there's Ashkenazi Jews, of which there's a population in New York, and also the Amish as well, which are in like Ohio and Pennsylvania. They tend to kind of, you know, breed within themselves, right? They have a large community, but it's still, and essentially all of the alleles that they have in their population were brought over by a limited number of individuals in the migration to the US, right? So that's kind of more population genetic stuff. But basically what it means is that if you don't have, and this is the same for dogs as well, if you don't have outbreeding, right, kind of new versions of alleles coming in, then by not outbreeding, you tend to concentrate those disease-causing alleles. And so as a result, you often get a higher proportion of certain diseases. Pardon me. So that's why, for example, with labs, they have bad hip problems, you know, later on in life. One of our dogs did. Also, founder effect. Yeah, I mean, I'm not an expert in population genetics, Caitlin, I have to say. I think they're all kind of terms which get at the same thing, right? So a founder effect is really, you know, you're taking a small number of individuals, they plop into, say, an island and kind of multiply, and yeah. So it's essentially talking about the same thing as a bottleneck. Bottlenecking can also occur for other reasons, though. So you can kind of be where you're at and have half of your population wiped out and repopulate, and that's still a bottleneck. So it's kind of, yeah, arm waving here, right? Because I have to wait till you get to evolution to learn the precise definitions. So interesting question, thing from Isaiah. Yeah, actually, the Jewish people, I guess that's the right way of putting it, have one of the best genealogy records in the world, equal, if not better, to the Mormon Church. And the reason for that is because they want to track who, well, partly for being Jewish, because you are Jewish through your mother, basically, but also for genetic diseases too. I guess we could manipulate our genomes to put genetic diversity back in, but yeah, you'd have to. I mean, really, outbreeding is not changing a few things. You're getting potentially a whole random collection of different alleles for different genes, and that's going to vary for whichever individual is coming into that population. So outbreeding is a much more thorough mixing of the gene pool, essentially. Yeah, bio too. But I mean, it's all part of the bigger picture, you know, it's all part of the same thing. It's just different ways of looking at it. What we're doing in genetics is really giving you the kind of foundation type stuff so that you can go off in like all those different directions and understand what's going on better, I guess is probably the way of looking at it. Actually, often in everywhere else that I've taught or taken genetics, population genetics is always part of the course. Here, for some reason, I don't know where this originated, but the population genetics is split off into another course and called evolution, and obviously with evolution too. Don't know why it's what whoever set up the program decided was what they wanted to do. So otherwise, normally you would be doing it as part of genetics. Okay, so monohybrid and dihybrid are two formal crosses. right? These aren't things that go on in like, real life, but they're very much theoretical thought exercises, both to learn genetics and both to test assumptions as well. And a hybrid is essentially a heterozygote. Well, it's kind of, yeah, that'll do. And finally, mono and dihybrid crosses have a very formal, stylized almost format. They never change. If it's this particular thing, it's a monohybrid cross. If it's not a monohybrid cross, right? And so part of the monohybrid and dihybrid crosses is they have these 3 generations. The P1 or parental generation, which are the true breeding parents. So one is homozygous dominant, one is homozygous recessive. The F1 or filial 1 generation are all heterozygous offspring of those parents. And then these are selfed to create the F2 generation. These are the individuals that have those ratios 3:1 or 9:3, 3:1 in dihybrid that are stereotypical of mono and dihybrid crosses. Now, before we get into Mendel and co, I want to just type in some assumptions. So Mendelian genetics rests on some assumptions. And so these assumptions are actually, when I type through them, we kind of go through them, you'd be like, that doesn't sound much like real life. And that's fine, because what we're doing here is we have a list of assumptions. And one of the powerful things about mono and dihybrid crosses or Mendelian genetics is you can test those assumptions. And so we always know what we're going to get from a dihybrid cross. 9331, always. So if we don't, that means one of our assumptions is wrong. Then you can figure out which one it is, right? So really these mono and dihybrid crosses are a somewhat artificial kind of constraint that we're looking at to see whether or not what we're interested in fits that very narrow type of cross, essentially. And if it doesn't, then that means that something interesting is going on. So the first assumption is that the alleles segregate randomly into gametes. And that's the law of segregation. So if you have a heterozygous individual, 2 different alleles, which gamete gets which allele? Totally random. And so you'd end up with basically 50-50 ratio of gametes with this allele to gametes with this allele. Two. What allele a gamete gets for one gene has nothing to do with oh, that should be which. Bad grammar. Which allele it gets for another gene. And this is the law of independent assortment. So basically it's saying if you have 2 different traits, each controlled by a different gene, then those 2 genes essentially act independently. This one, if it has two alleles, right, you get 50-50, this one and this one. And if you get this particular allele, dominant allele, for example, you could get either the dominant or the recessive allele for the other gene. Right, we'll actually spend a lot of time showing how that doesn't necessarily work that way later on in the second section during linkage. Right? But this is our assumption, right? So those are basically, one is all about monohybrid cross. Two is all about dihybrid cross. Right, so those are the two crosses that are used to illustrate that principle. Now, I'm spending, you're probably thinking, oh man, when are we going to get to Mendelian genetics? We'll get there, I promise, right? And it's actually pretty straightforward and simple once we do. But this stuff is really important to get, because if you don't get it, all that stuff is just like, oh yeah, sure, whatever, right? You won't really understand it. If you don't understand it, then the stuff that we do later on, where we go, well, that isn't right, you know, let's see if we can break this, then that's not going to make any sense, right? And the stuff we do later on is actually far more interesting, which kind of probably shouldn't say that as a professor, but anyway, more interesting, but also more complex as a result, right? So it's good to get this stuff down ahead of time. And then we've got some basic stuff. Reproduction is random. So who mates with who is totally random. Which gamete fertilizes which gamete is totally random, right? So all random, which, you know, clearly isn't real life. I mean, I didn't reproduce with my wife just randomly, bumped into her in the store, and there you go, got kids. You know, there's a little bit more to it than that. So clearly that's not a 100% true assumption, right? In many cases, fertilization actually is mostly random, but not always either, right? So anyway, just give you an example. What else? No mutations, which always makes me think of the no capes scene in The Incredibles, which is one of my favorites. I can never remember the last name of Edna though, for some reason. Anyway, and also no selection. So there are all combinations of alleles and all phenotypes have equal fitness, right? If you had a situation where one particular genotype resulted in that organism dying or that individual dying, then obviously the results of your cross are going to be skewed, right? They're not going to fit the pattern that they should, right? So these are really, really important assumptions, right? This is what we kind of, what the monohybrid and dihybrid crosses are completely based upon, right? And obviously they're not assumptions which really stand up in real life, right? There are very few situations where this would be true, but that's what makes them so useful, right? Because then we can see which assumption is not holding up, right? And from that, we can learn interesting stuff. I put a water bowl down, but she hasn't drunk from it. Okay, all right, not too much longer left, only 8 minutes, and you can enjoy the rest of your day. So essentially, well, we've got at least another couple of, I always have to check the schedule because, as I say, memory like a fish, and I'm also teaching 2 versions of this too. Ooh. All right, well, we might run over a little bit. Oh, yeah, right. I'm trying to read my own schedule that I wrote. Okay, we're all doing fine. Quite often, I'd rather spend more time on like the basics and the foundation stuff because that'll allow you, even if we don't cover things as thoroughly as we can, it'll allow you to figure that stuff out too. Anyway, Mendel, smug mug, kind of handsome chap. Big deal about Mendel really was that he did two things. One, obviously, he determined that inheritance was particulate, right? And by that, essentially, before Mendel came along and did his pea experiments, there was this big debate in genetics. Well, genetics as a field didn't even exist then. Big debate in natural philosophy is actually what the natural philosophers. And the debate was, is inheritance blended or particulate? Right. So are there discrete factors that determine phenotype, what you see? Or is it something that you just kind of like smoosh together the two different individuals and you get something in between? Right. And so we now know that inheritance is down to genes and alleles and things like that. They didn't know any of that, 170 years ago. So he actually showed that it was distinct, discrete factors that control traits, right? Even if though he didn't know what those were, he was able to kind of figure out the mode of inheritance and the like. The second thing, which is arguably more important in many ways, is he actually pioneered the scientific method, right? He kept notes, he recorded data, he analyzes data, he had a hypothesis, right? He tested that hypothesis, and his findings were supported by data. Up until that point, it was really about who could shout the loudest, right? Or who had the biggest beard or whatever, right? In many ways, well, this is somewhat heresy, right? So don't tell anybody. Darwin was in that camp, right? So Darwin, even though he's kind of lionized and is like the patron saint of biology, he just made a bunch of observations, right? He didn't actually carry out any experiments. He was a natural philosopher, right? And so he was in some ways right, not 100%. And he was obviously a very clever chap, but he still didn't actually test any of his ideas. They were just philosophies, essentially, right? Mendel was the first one to actually do experiments to test those ideas, to test his ideas. And he used peas. Peas are arguably the most boring thing to talk about when it comes to genetics, because they're either round or they're wrinkly or whatever, but it's what he used. Part of the reason was because they grow relatively quickly. I personally use cats. Cats are way more interesting, but they take a long time to grow. And can you imagine having enough cats to get data to figure out a monohybrid or dihybrid cross? That'd be a lot of cats. Anyway, so peas are what we have. And I'm not going to cover all that stuff because that's not super interesting. Essentially, that leads us to the monohybrid cross. Okay, so we'll go through this briefly today, and then we'll go through in more detail on. Actually, what we'll do is we'll cover this today as is on your screen, and then on Monday we'll go through it on my whiteboard. Ta-da! It's really cute. Yesterday, my puppy kept video bombing the Zoom session. She would just kind of lie out in front of my whiteboard and stop me doing my work, which is very funny. Anyway, so monohybrid cross, three generations, right? P generation, F1 generation, P generation, and F2 generation. P generation are two homozygous individuals. You don't necessarily know at that point which one of these is dominant, because one of them will be homozygous dominant, the other one will be homozygous recessive. You don't necessarily know which is which, right? You then cross those together. That gives you the F1 generation, and now these are all heterozygous, right? So if these are heterozygous, they're going to express the dominant allele, correct? So which of the parents, and this is a question I'm asking you lot, which of the parents is a homozygous dominant genotype out of the round versus the wrinkled? Which parent is homozygous dominant? Yeah, the round. Why, Bianca? You're correct, 100%. Why are the round ones the homozygous dominant? And it doesn't have to be Bianca answers. Sometimes it's a little bit of a punishment in a way of answering the question correctly, right? You just get another one. So look at the, yeah, that's it, exactly. Good stuff, right? Because it's the trait that shows in the offspring, right? And as these offspring are heterozygous, they will express the dominant allele, right? And so even if you don't know what the genotype is in the parents, you can figure it out based on what phenotype the F1 generation has, right? Okay, so that's one thing that's worth remembering, right? You can always figure out the genotype of the parents by looking at the offspring, at least in a monohybrid cross. And then these are crossed together, and because they're heterozygous, they each have two different potential alleles, which when you combine them together will give you, actually it's worth pointing out this. You'll get 1 to 2 to 1 genotype ratio, but because, yes, exactly. Get back to that in just a second. Let me finish typing this out, Rebecca. But because the first one and the two both have dominant alleles, and we'll get to this in the Punnett square on Monday, they all have the same phenotype. So that's why you have the 3 to 1 phenotype ratio. Now, the good question from Rebecca, this is roughly 3 to 1. Now, because of just biological noise, you're never going to get exactly 3 to 1 dominant to recessive, but it should be pretty close. We'll actually get into how you can tell statistically whether that's close enough or not, right? We'll do that in the probability and statistics stuff next week, right? But it should be approximately 3 to 1. If it's not, something up here is not right, right? One of your assumptions is wrong, or you didn't have what you thought you had in terms of genotypes. It should always be a roughly 3 to 1 ratio. Cool. All right, that's me done for today. Thank you for all the fabulous questions. Really does make it a lot of fun teaching. And hopefully we more or less stay on track and cover the stuff that we're supposed to. Okay, so I would strongly suggest you have a look through this PowerPoint if you haven't done so already, mostly at the dihybrid stuff, the test cross and the dihybrid. Those are things that trip people up, and they're also things that you're going to get tested on a lot, pardon me, in the exam, so they're worth really getting down. So have a look at those, and then we cover them on Monday. They'll be a little bit more accessible. In the meantime, have a lovely day, enjoy your weekend, and I'll see you on Monday.